Hello. Sorry for being a little late. I had a little trouble logging in. Can somebody confirm that you can hear me?
I can hear you.
Great.
Yes.
All right. Let's begin. Come on. So today is February 21st, and we are supposed to be working on Chapter 8 and finishing it. Let me share my screen here. All right. So we should be working on Chapter 8 and finishing it. I don't remember how far along we are in Chapter 8. And starting a little bit of Chapter 14, if we can. In the lab today, we're going to be having Lab 9. And of course, there's the unknown work. Any questions about what we're doing? All right. Remember to work on your unknown project because this project will take you some time to finish. There might be one or two students who have finished the project now, but the majority of students are still working on it. And some students have, we better be further than this. Let me see. Because we're not going to finish it if we're there.
The last note I have is spontaneous mutation.
Okay, yeah, I've got mutation right here. Thank you. Let's see if we're going to finish this today. We have a number of slides to go, but we probably will finish Chapter 8. Come on, come on. All right, if there's no questions about what we're doing, let's go ahead and get started. Remember to work on your unknown project. If you have questions about the unknown project, please email me and I'll try my best to help you. Some people have not done a thing with the unknown project. And like I said, if you've got questions about what to do, just email me and I'll try and help you. I will not give you what your unknown number, what species it is, so don't bother asking that. And I will not tell you how to read a test. You should read the instructions on the different tests to find out what the test is doing. Of course, you should do it to find out how long the test incubates and then how to read the test. You should read that on your own. All right, so we were talking about mutations, and I'll remind you there's a mutagen that can increase the rate of mutation. So it's some chemical or physical agent that increases the mutation rate. And then there's a spontaneous mutation, which occurs in the absence of everything. It just happens when a mistake in the DNA gets made, like when the DNA is replicating. There are different types of mutations. There's a base substitution where one base is substituted for another, and this is called a point mutation. Point mutations can be one of three types. It can be a silent mutation, a missense mutation, and a nonsense mutation. And we'll talk about those in just a minute. Besides the base substitution, you can have a frame shift mutation, and that is where not where one base is substituted for another. This is base substitution. It's where a base is inserted or one is removed from the DNA. And it could be a multiple base is inserted or a multiple base is removed. It's called a frame shift mutation because when you add an extra nucleotide, it will shift the codons from that base, and we'll look at that in a little greater detail. There is an exception for the frameshift mutation, and that is if you add 3 bases or subtract 3 bases, it's not a frameshift mutation, it's an adding or an addition, and it's removing 1 codon or adding 1 codon. And then lastly, there's a macro mutation. You should pay attention to my presentation and my notes, because for some reason, a macro mutation is not discussed in the book. It is where an entire region of a chromosome is either added or deleted. And your book does not discuss it, and I'm not sure why they don't discuss it. It is a mutation, but it's a very large mutation in the DNA where many nucleotides are changed. And it's not all that uncommon. We'll talk a little bit more about that. There's a boy who possibly is a mutant, and you can see, let me see if I can blow that up. Spoons are sticking to him, like he's a magneto. Whoops, went too far. And people were wondering, does he just have sticky skin or is he magnetic? And they were doing a study on him. I read that they were studying him, but I never got the results of the study, so I don't know what the finding was. All right, a base substitution is where 1 base is substituted for another. Let me blow this up. Such as when GC, come on, mouse. My mouse has died. There it is. GC gets substituted for a GT. Then when this replicates, that strand will, dang it, that strand will actually replicate okay, but this strand will not replicate okay, and that will fix the mutation. Now we have a TA pair in here where it should be GC. And that will, in this case, cause a change in the amino acid because it's a different codon. Anyways, that's the base substitution where 1 base is substituted for another. This is also called a point mutation. The mutation can be neutral, where the mutation doesn't change the benefit for the mutant individual. So this individual has no benefit over the normal individual. This could be a benefit. And I think I talked about that, where there was a mutation in somebody who was breeding fruit flies. And in the, well, we got to do it this way. Maybe I can do a clear one, partially clear. In the fruit fly container, the fruit flies would put their pupa on the sides of the container, and the bottle was full, so there was no more room for the pupa. And there was a mutation that occurred in the population, and now the fruit flies could lay the pupa or put the pupa on the top where the cotton was. And that was just a cotton swab, not a swab, but cotton plug to cover the container so the fruit flies couldn't come out of the container. And the thing is that that mutation was beneficial because now the fruit flies that could have that mutation could put their pupa on the cotton swab, and then they were increasing in the population, whereas the other fruit flies were not. Any question about that? And then most mutations are harmful, in which case the mutant individual is less likely to survive and reproduce. And an example of that is Tay-Sachs disease, where the mutant has less likely to live and reproduce. Any questions about that? Yep, here we go, a silent mutation. Let's talk about a silent mutation. This is a mutation where the DNA is changed, but it does not result in a change in the protein. So the DNA base is changed, but the amino acid is not changed. That would be something like UUU is changed to UUC. It's the same, well, it's a different codon, but it codes for the same amino acid. So the protein is unchanged. Come on, mouse. And that's a silent mutation. There's no effect on the protein function. Any questions about any of that? A missense mutation is where you change one base and it results in a change in the amino acid. For example, we have AAA and, whoops, I guess it's not AAA. What the heck is the AAA? Isn't changed. It's the CCG, which is changed. Come on, mouse. So this region right here is changed. And it's changed to TCG. That changes the amino acid from glycine to serine. This is called the missense mutation, where you change one nucleotide base and it results in a change in the amino acid in the protein. Any questions about a missense mutation? Once again, this could be a neutral mutation. It could be a beneficial mutation. Most likely it'll be a harmful mutation. A nonsense mutation is where you change one base. So what are we changing here? TAC. Right here, TAC. And then we change from TTC to ATC. Where you change that base, it results in a stop codon. So instead of coding the amino acid lysine, we're now coding stop. And that will result in a truncated protein. So normally the protein is 4 amino acids long, which is a really short protein. Most of the times they would be, gosh, over 400 amino acids long. But the point is it's truncated, and in this case, we have a smaller protein. This often results in a protein that doesn't have function. It really depends on where the function in the protein is. If it'll lose its function, like I said, oftentimes a nonsense mutation will result in a protein that does not function, but there are exceptions. Any question about any of that? All right, a frame shift mutation is where you unseed or delete one base or a multiple of bases, except for a multiple of 3. I'll explain the multiple of 3 in a minute. So we have AAA here, coding for the amino acid phenylalanine. And we remove one A here. Now the codon is going to be, it'll read the AA, and it'll actually be UU. But you see, it doesn't have the third A here. And so it takes the next nucleotide. And UG would be the next amino acid. So from this point on, it's a frame shift mutation because all the other codons downstream of this will be altered. So initially it was supposed to be methionine, lysine, phenylalanine, glycine, and then stop. And now it's methionine, lysine, leucine, alanine, and the stop codon isn't red because you don't have UAA anymore. What are we going to have? UUG, GCU. We have AA something, and it's not a stop codon. The point is that the frame shift mutation changes the protein from the frame shift, the point where the frame shift is. Come on, mouse, which would be in this case, right here. This happens whether you add a base or delete a base, you get a frame shift. It also happens if you add 2 bases or delete 2 bases, or add 4 bases or delete 4 bases, or anything but a multiple of 3. If you remove 3 nucleotides, like let's say you remove UUG, UUU, what you do is you remove this amino acid, and then it'll be methionine, lysine, and then glycine, and then stop. So it's not a frame shift. You have altered the protein because we removed 1 amino acid, but it's not a frame shift because only this 1 amino acid has changed. All the others are the same. So a multiple of 3, either a deletion or an addition, is not a frame shift. Is that clear? Everyone good with the frame shift mutation?
Yeah.
I'm sorry? A frame shift mutation often removes the function of the protein because a large part of the protein is changed. But once again, it depends on where the function of the protein is. If it happened to be only at the start of the protein may have function. All right, so that's it for mutation. So let's talk about the causes of mutation. Oh, I forgot to talk about a macro mutation. So let me come back here and talk about a macro mutation. A macro mutation is where an entire region of the chromosome is either removed or added. One that's actually quite common in the human population is Down syndrome, which has an extra chromosome 21. Most of the times, if you add an extra chromosome, it'll be lethal and the fetus will have a spontaneous abortion. But with Down syndrome, because that chromosome is pretty small, it is not lethal, and the individual will be born. And that'll result in a Down syndrome patient. Any question about that? Anyways, that's one example of a macromutation. All right, let's talk about the causes of mutation. Mutations can be spontaneous, where they just happen because the DNA is being replicated and the DNA polymerase makes a mistake, in which case the mutation can then happen. There is a possibility that the cell or the system, whatever you want to call it, will catch the mutation and then correct it. We're not going to talk about any of those corrections, but cells do have like a proofreading function, and they frequently can catch a mutation and then correct it. And what they do is they know which strand of DNA is the original strand and which is the newly replicated strand. And the cell assumes it's the newly replicated strand which has the error, the mutation. And most of the times that is correct, okay? Especially if it's a spontaneous mutation. And so it reads the original strand of DNA to repair the newly replicated strand of DNA. And the second cause of a mutation is a mutagen. which is an agent which increases the mutation rate. There's two types of mutagens. There's radiation and there's chemical mutagens. With radiation, there's two types of radiation: ionizing radiation, which is a radiation with a high energy component. And when this radiation hits an electron, it will give that electron enough energy to leave the atom. So that will create an ion that is left behind. And then this ion can react with DNA, causing the mutation to the DNA. Any question about that? It's also possible that the radiation can hit the DNA, causing the DNA molecule to lose an electron, and then the DNA will be an ion, and it can react with other DNA. Another region of the DNA, I guess I should say. There's also non-ionizing radiation, that is UV light or ultraviolet light. What happens with UV light when it hits DNA, it causes two thymines that are next to each other to form a dimer between them. Let me see if I can blow this up. So two thymines that are next to each other, when the UV light hits it, causes a dimer. That's two covalent bonds between the thymine. This thymine dimer doesn't pair correctly with the corresponding strand of DNA. In fact, there will be no hydrogen bonding between these thymine dimers and the A in the corresponding strand of DNA. It also causes a bubble in the DNA over here, and DNA replication and DNA transcription cannot go past the thymine dimer. So this will stop DNA replication and DNA transcription. We won't talk about how cells could repair thymine dimers. For one thing, this doesn't happen, light repair in what do you call it, non-marsupial mammals, meaning most mammals. But the thymine dimer does cause that's essentially a mutation in the DNA, because the T's are not responding correctly like a normal T. They're not binding, hydrogen binding to the other pair, and then the T's cannot be read by DNA replication or DNA transcription. Any questions about non-ionizing mutation? All right, there's also chemical mutagens. We're only going to give you an example of one. Nitrous acid is a mutagen. When nitrous acid gets on DNA, it chemically changes the adenine. So there's nitrous acid getting on the DNA. I guess the DNA here, sorry. It chemically changes this A. So it is an A in a sense, but it's a little bit modified. And this A now no longer binds to T. And when the DNA polymerase reads this modified A, it doesn't read it as an A, putting in the T. It actually reads this A as a G, and then it will put in a C. This will cause a mutation because when this gets replicated, that'll be the wrong strand, GC, as opposed to AT. And then this AC will, of course, read again as AC because that A is going to be read as a G. Anyways, that is obviously a mutant cell here. Come on. This is a mutant cell here. This one is read like a mutation. That A is an A, but it's chemically altered, and so it's read like a G. Any questions about how nitrous acid acts as a mutagen? All right, the spontaneous mutation rate is for every 1 times 10 to the 9th bases that are replicated by DNA polymerase, 1 will have a mutation. So that means 1 base in a billion will be mutated. Now, there are about 1,000 bases in a gene, and so that means the mutation rate for a gene will be 1 in 10 to the 6, or 1 in a million replicated genes will be mutated. Any question about any of that? These are very low numbers, but when we're talking about the human population, it's something like it's close to 8 billion people. And so that means that in the human population, for this last generation, there were about 8 people who had a replicated base that was a mutation, meaning that of these, 8 billion people, there's about 8 of them that are mutants by spontaneous mutation. Any question about that? And then when we're talking about genes, it's even higher because, like I said, there's a lot of genes in an individual. And the population is like 8 billion, and you only have a spontaneous mutation rate. 1 gene is mutated in 10 to the 6 replicated genes. And I used to know how many genes were in an individual. It's nowhere near a million. It's something like 40,000, something like that. So in an individual, you're unlikely to get a mutated gene, but in the human population, you are. Any questions about any of that? All right. Mutations increase the mutation rate, and they increase the mutation rate from 10-fold to about 1,000-fold. And that means the replicated genes will be mutated for 1 mutation in either 1,000 genes or 100,000 genes, 10 to the 5th genes. Any question about any of that? So when mutagens are around, the mutation rate is much higher, and the number of mutants will also be much higher. Do you guys know what the most common mutagen is that humans are exposed to, besides maybe sunlight? Anyone know? What's the most common mutagen that humans are exposed to?
You said not UV?
Not UV. Obviously, UV from the sun is a mutagen. And so I guess that would be the most common mutagen. And our skin cells are mutated all the time, but our skin is fairly good at repairing those mutations. But my point is, other than the sun, what's the most common mutagen that people are exposed to? Anyone know? Nobody are smokers? Smokers should know this answer. And that's cigarette smoke is the most common mutagen that people are exposed to. All right. Identifying mutants experimentally. You can test for a mutant with 2 ways. You can have positive direct selection or negative indirect selection. A positive direct selection means you can detect the mutant cells because they grow or they appear different. Like if this were to walk down the streets in the United States, people would look at it and say, yep, there's a mutant, right? Because the individual looks different. A negative indirect selection is you detect the mutant cells because they do not grow. under normal circumstances. Negative selection is actually something that I did when I was in undergraduate. And one way to do it is you get a Petri dish containing isolated colonies, and you get a velvet stamp. You put the stamp into the colonies, and then the cells from the stamp stick to the velvet, which is shown here. And then you stamp different Petri dishes from your stamp. And then the cells will be transplanted to the Petri dishes, and then you grow up the colonies. This colony is a mutant because it's growing on media that contain the amino acid histidine, but in media lacking the amino acid histidine, this colony cannot grow. And that would be from probably that stamp there, this colony here. Any questions about that? So this is a mutant, and it is a mutation for needing the amino acid histidine, meaning this amino acid is not made by this colony. And that's why this colony doesn't grow in this media. So you have to supply the amino acid, and then we can get it to grow. Any questions about negative selection? All right. That's it for mutation. Any questions about mutations? If not, we're going to move on to recombination. Recombination is an exchange of genes between two DNA molecules. It doesn't create new mutations, but it shuffles the genes around so that we can have different combinations of genes. Is that clear? Like here we have the father's chromosome, the blue one, lowercase alleles, and there we have the mother's chromosome, uppercase alleles. You can have recombination where part of the father's chromosome breaks off the father's and anneals to the mother's, and then part of the mother's chromosome breaks off the mother and then anneals to the father's chromosome. And now we have on the father's, what used to be the father's chromosome, lowercase e, lowercase f, lowercase g. But, whoops, gotta come down here. I'm not seeing my move down thing. Why is that the case? I'll just do it manually and hopefully I don't do it too far. I think I did that too far. Let's shrink that. There we go. So lowercase e, lowercase f, lowercase g, and then uppercase h from the mother's chromosome, uppercase I from the mother's chromosome. This will put lowercase g and uppercase h together. And so recombination reshuffles the gene, makes new combination of pairs possible. Any question about any of that? Crossing over is when two DNA molecules come together and then break and exchange pieces. Whenever crossing over occurs, there will be a recombination. Come on. I don't have my mouse for going down, but I have it for going up. Do you notice the difference between recombination and crossing over? They are similar. Crossing over always has a recombination, but recombination is a little more general. Recombination is the exchange of genes between two DNA molecules. Crossing over is physically the coming together of two chromosomes, and then they switch pieces. Okay. Can recombination ever occur without crossing over? Yes. For example, you can have recombination where a portion of, let's say, the mother's chromosome comes out of the mother's chromosome. Let's make it capital G. And then it joins with the father's chromosome. And now we have a lowercase g and an uppercase G on this chromosome. Okay, so that would be recombination, but not crossing over. Any question about any of that? The point is, crossing over always involves recombination. Recombination usually involves crossing over, but it doesn't have to. Recombination is a little more general than crossing over. Okay. Any questions about recombination and crossing over? I guess before I go off of that topic, we should, I'm going to talk about it in a little bit, say that all new genetic variation in a species or in a population has to come about by mutation. Spontaneous mutation or mutagen causes it, meaning that recombination doesn't cause new genetic variation, only mutation causes new genetic variation to arise within the population. Let me shrink that down. Let's talk a little bit about genetic transfer. Vertical gene transfer occurs during reproduction. So between generations of cells, one parent cell will divide into two daughter cells, or if you want to talk about the human case, two parents will reproduce and have an offspring. That is vertical gene transfer. Okay? Horizontal gene transfer is the transfer of genes between cells of the same generation. So genes leave a donor cell and the genes are taken up by a recipient cell. That is horizontal gene transfer. Horizontal gene transfer can occur in three ways, at least in bacteria. In humans, well, you can have transformation happen in humans. You don't really have conjugation happen in humans. You can have transduction happen in humans. So it happens in two ways in humans. And we'll talk a little bit about that. Transformation is where DNA comes out of a cell. The DNA is naked DNA in the environment. And then that naked DNA is taken in by another recipient cell. And then part of the naked DNA is incorporated into the chromosome of the recipient cell. This can bring in new genes to the recipient cell. For example, on the naked DNA, we have lowercase a gene. You don't have lowercase a in the recipient cell. And then the DNA is brought into the cell. There's a recombination that happens where the DNA from outside the cell recombines with the chromosome. And then we now have the lowercase a gene being inside the chromosome right there. And we're not going to talk about how this happens, but the lowercase a recombines with the uppercase a. The recipient cell loses the uppercase a gene, but gains the lowercase a gene. And that's much more complicated than I want to talk about. I just want to mention that the lowercase a gene from the naked DNA comes into the cell, and then that lowercase a joins with the chromosome, bringing this cell new genes. Now, if this lowercase a is expressed by this cell, we will now have a new gene being expressed by this cell. And this is important because if lowercase a, for example, codes for antibiotic resistance, this cell will now be antibiotic resistant. Okay? Any question about any of that? This process of taking in DNA from a donor cell, which that cell usually dies, by the way, while the DNA is naked DNA in the environment. And then that naked DNA is taken up by the recipient cell. All of this is called transformation, the process of transformation. However, this cell, once it expresses a new gene, we can say that this cell is transformed because it can express a new phenotype. Meaning if lowercase a codes for antibiotic resistance, this cell will now gain the property of antibiotic resistance. Any question about any of that? So yes, you do need to know that transformation means two things. It means the literal process of taking up DNA by a recipient cell. And it also is the change in the phenotype of this cell. That's also called transformation. We have a picture of the actually the transformation process and then the phenotypic change of the cell, where the DNA is seen by the cell, the DNA is taken up by the cell, it then gives the cell a little indigestion, and then the phenotype of the cell is transformed. Any question about any of that? All right, that's transformation. The second way that new DNA can be brought into a cell is by conjugation, sometimes called sexual conjugation. If in conjugation we have, well, there's two types. There's the gram-negative cells. They have a sex pilus, which is this very long, I don't know, tube-like thing extruding from actually this cell. And then in gram-negative cells, we have a mating bridge where the two, not gram-negative, gram-positive cells, we have a mating bridge where two cells actually come together and their cell membrane fuses right here. And then instead of the cell membrane coming this way, it then goes around so that these two cells are connected, and the cytoplasm from this cell can move into that cell, and the cytoplasm from this cell can move into that cell. This can allow genetic exchange, meaning DNA, especially plasmid from this cell, can move into that cell or vice versa. With the sex pilus, the DNA always moves out of the cell that has the sex pilus. Let me blow this up. So this cell encodes the sex pilus, and there's an F plasmid, meaning a plasmid in this cell. That plasmid, why it's called the F plus plasmid, is this plasmid codes for the sex pilus. It has the genes that make the sex pilus. This cell literally searches for an F minus cell. That's a cell that doesn't have the F plus plasmid or the sex pilus. And the sex pilus can literally skewer the F minus cell. And then that sex pilus can act as a winch, pulling the two cells closer together. They don't have to be this close together or this close together, as it's shown here, because this sex pilus is actually hollow. And then the F plus plasmid will replicate, so there will be two copies of it, and then one copy will move through the sex pilus into the F- cell, which is shown here. Although this has the two cells brought very close together by the sex pilus. The F plasmid replicates. One copy moves into the F- cell. This F- cell now has the F+ plasmid in it. The F+ plasmid will code for a sex pilus. So this cell will now grow a sex pilus. Come on. Sorry, my mouse is dying. Come on. This cell will grow a sex pilus, and now we call it an F+ cell because it will go and look for an F- cell, and the sex pilus will literally skewer it, and then the two cells will be winched together, and the F+ plasmid from this cell will be replicated and move into the F- cell. Any questions about conjugation or sexual conjugation? All right. If you're wondering why this is called sexual conjugation, you can ask your significant other why this is called sexual conjugation. Any questions? Let me state once again that sexual conjugation is one way where genes from one cell can move into another cell, changing the phenotype of this cell. So the F plasmid moves from this cell to that cell. That can change the phenotype here, and it can give the cell a new phenotype. I haven't stated it, but we can't have other genes on this F plasmid. And let's say that this cell is antibiotic resistant, but this cell is antibiotic sensitive. If the antibiotic gene is on the F plasmid, it will then move into this cell, and then this cell will be antibiotic resistant as well as this cell. All right, the third way that genes can move about in a population is by transduction. There are two types of transduction. We're only going to talk about general transduction. General transduction uses a virus, and we haven't talked about viruses yet, so let me briefly talk a little bit about viruses to explain what's going on. The virus will infect a bacteria cell. We're talking about a bacterial virus. And when it infects the bacteria cell, it'll inject its DNA into the bacteria cell. The virus wants to run the show in the cell, so the first thing it'll do is degrade the host chromosome. Why? Because if the host chromosome's gone, then only the viral DNA will be around to run the show in the cell. And what the virus will do is use this cell to replicate itself. So it will replicate the viral DNA, transcribe the viral DNA to make viral messenger RNA, and then translate that to make viral protein. And then the viral protein will get together with the viral DNA and then replicate and make, that makes more viruses. So the goal of the virus is to infect this cell and then use this cell to replicate the virus. Okay? And when the virus is replicated, it will then lyse the cell to release the newly replicated viruses. Okay? But occasionally with viruses, mistakes are made, partly because viruses have so many offspring, they're not worried if one of the offspring is a mistake. The mistake happens with, let me see if I can see it. I'm not seeing very well today. Oh, that's better. You'll notice that the chromosomal DNA is bluish and the viral DNA is reddish purple. When the virus is replicated, it just puts the DNA into the virus protein cap, and then these are all normal viruses. But you note that one and that one did not get the viral DNA. Instead, a piece of the cell's chromosome got inserted into the virus. Now, when this virus replicates, it will infect this cell and, like the normal process, inject its DNA into the cell. However, this is not viral DNA. It's DNA of this chromosome. This DNA can then recombine with the recipient cell's chromosome, bringing in new DNA to this cell. And this DNA came from this cell. And that's another way that cells can pick up new genes. For example, if this cell was sensitive to antibiotics, like penicillin G, and then this piece of DNA got inserted into that virus, so it's here, and then that virus incorporates into the viral DNA, incorporates into the host recipient chromosome, we now have the gene for penicillin resistance being brought to this cell. Any questions about any of that? Anyways, that's transduction in bacteria. I've thought about transduction, and I think transduction happens all the time in people's nose cells, meaning when we get the cold and the flu. A virus can replicate and mistakes can get made, and then a piece of the host cell's DNA could be put in the virus, and then this virus can go to somebody else's nose and then bring a gene to somebody's cell in the nose. Okay? So I think transduction happens, at least to human nose cells, maybe throat cells also, all the time. But who cares, because that mutation will be one cell in the nose or in the throat of the individual, and that mutation will never be passed on to that individual's offspring. Okay? So I think transduction does happen in the human population. It's just that it's not important because it doesn't usually involve a germ cell, meaning a germline cell, sperm or egg. And it would only be important to the human population if it did affect the sperm or egg cell. Sexual conjugation, I don't think happens in the human population, where we have cells exchanging DNA with the plasmid. For one thing, humans don't even have plasmids. And even a mating bridge, like I said, this just doesn't happen in human cells that I'm aware of. Transformation does happen in human cells. The difference is our cells aren't very good at taking up DNA, especially DNA that's in the environment. But you could do this in the lab, where we put human DNA with a human cell and then inject this cell, which incorporates that piece of DNA. We inject that back into the person. That could be done in the lab. But like I said, in reality, transformation very rarely would ever happen in the human population. But in bacteria, it happens all the time. Now, let me talk about how often this happens in bacteria. If you remember, the spontaneous mutation rate in Oh, let me see if I can get this. Here I can go. The spontaneous mutation rate will be one gene will get mutated in 1 times 10 to the 6. Now I'll just go 6. 6 genes. Right? So that's the spontaneous mutation rate. So we'll go SM for spontaneous mutation. That should be a capital S. Transformation in bacteria happens about 1, I'm just going to copy this, 1 in 10 to the 5th times. What the heck? Ah! I thought I copied that. Better go there first. Conjugation in bacteria also happens 1 in 10 to the 5th times. And then transduction, oops, gosh. Also happens about 1 in 10 to the 5th cells. So transformation, conjugation, and transduction in bacteria all happen about 10 times more frequently than spontaneous mutation. Any question about any of that? This is an important part to know. And the other thing is all new genetic variation in the species or in the population has to come about by spontaneous mutation. However, once it exists in the species or the population, that mutation can be spread around by transformation, conjugation, and transduction. And so the next cell is most likely going to get it from one of these rather than get it from spontaneous mutation. Any questions about any of that? Hopefully that's clear to all of you. All right, let's close this down. Good, just checking to see if someone was there, and you're so quiet, I wasn't sure. Let's go back to our lesson. All right. So if a new genetic variation appears in the population, it's most likely to come from transduction, transformation, or conjugation, as long as the variation exists in the species. If it doesn't exist in the species, it must come about by spontaneous mutation. And I did mention with humans, a spontaneous mutation did happen about 10,000 years ago, where the humans, all the population had brown-eyed alleles, and then a blue-eyed allele spontaneously mutated in Northern Europe about 10,000 years ago. So that's actually an example. All right, let's talk about plasmids. We've talked a little bit about them. They are small, circular, extrachromosomal DNA. They do contain genes, but unlike the chromosome has tens or hundreds of thousands of genes on it. The plasmid only has a few genes. Like this one here, this plasmid has 1, 2, 3, 4. I can't read that. That could be 5, 6, maybe 6 or 7 genes on it. Okay? So there's only a few genes on a plasmid. A plasmid is much smaller than the chromosome, and the plasmid does replicate independently from the cell's chromosome. In fact, we saw that independent replication when we were talking about the F plasmid. Here, the F plasmid replicated independently of the chromosome, and then one copy moved into that cell. The chromosome did not replicate in this situation. So the two are replicating independent of each other. Any questions about plasmids? Plasmids are important because they are more likely to spread from one cell to another cell than the host chromosome. It is possible for the host chromosome to move, like in conjugation, but most likely it's a plasmid that's going to move in conjugation. And that's true for both the sex pilots, the gram-negatives, as well as the mating bridge on the gram-positive. The plasmid is much more likely to move from one cell to another with a mating bridge rather than the chromosome. Plasmids can recombine with the chromosome, or a gene from the chromosome can move into the gene of the plasmid, and that's one way that genetic information can move about in the cell and then between cells. Any question about any of that? You should know that there are different categories of plasmids, but you don't need to know the different types of categories. All right, the last genetic element I need to talk about is a transposon. A transposon is a genetic sequence, including insertion sequences. So this is a transposon, and it has one gene on it, a transposase gene, and then inverted repeat on both sides of it. This is called the insertion sequence. The insertion sequence, as well as the transposase gene, can cut out this gene from a region, let's say it's on the chromosome, and then both the inverted repeats and then the transposase gene can go elsewhere, like another place on the chromosome, make a cut in the chromosome, and then insert the transposon into that cut. Okay, so that's what transposons do. They are genetic regions of DNA that can move from one region of the DNA to another region. So they can move from one region to another. And they're set up to do this. The transposase gene encodes an enzyme that cuts off the DNA here and here. And I think it also cuts the new place where the transposon is going to move into. And then the inverted repeats help move the transposon into the newly cut DNA. And then the transposase gene reseals the DNA. Okay, so this transposase enzyme both cuts the DNA and then acts like ligase, resealing the DNA. Any questions about that? Simple transposons, all they have is the inverted repeats and the transposase gene, but complex transposons can carry other genes, like for this transposon, we have kanamyosin resistance on the transposon. And it's curious, when you have another piece of DNA, you actually have a simple transposon over here and a simple transposon over here, and then the inserted DNA between them. And then this can cut itself out of wherever it is on the DNA and then move to another region of DNA. What's important about transposons is they can move DNA from a chromosome to a plasmid or from a plasmid to a chromosome. Why this is important is we're talking about shuffling genetic information around in a cell and between cells. Well, transposons can move DNA from a chromosome to a plasmid. And then a plasmid can move from one cell to another cell. And that can move about DNA from a region of the cell to another region of the cell, same cell, and then from one cell to another cell by conjugation, for example. Okay, so transposons are very important in shuffling the DNA, much like recombination is. And then transposons also help the DNA literally move from one cell to another cell, because the transposon can move the gene, like kanamyosin resistance, into a plasmid. And then a plasmid is much more likely to move out of that cell and into another cell. Any questions about any of that? Oh, this is just showing you that kanamyosin resistance moved from the chromosome to a plasmid, and then this plasmid can move out of this cell and into another cell. So, genetic variation in the population, if it's brand new genetic variation, meaning it never existed before, then it has to come about by mutation. And that is the only way brand new genetic variation can come about in a species. And that's not entirely true. I guess genes from one species could move into another species, but you're getting my point, okay? And that is, if it's brand new genetic information, it has to come about by spontaneous mutation, okay? But if that genetic variation exists, then it can move about in the population by transformation, conjugation, transduction, and then crossing over and transposition. Transposons can help move this DNA around and make new genetic combinations of genes. The point is, genetic variation can come about by all of these forces: mutation, crossing over, transformation, conjugation, transduction, and transposition. All of these forces of genetic variation can drive evolution, and then evolution will occur via natural selection. And this explains genes and evolutions, that genes evolve by new genes being created by spontaneous mutation, the genes moving about in the population through transformation, conjugation, and transduction, as well as, I suppose, by new spontaneous mutation, because you can have, in the population, you could have two mutant individuals, both of them getting it by spontaneous mutation. And then that will drive evolution, which will proceed by natural selection. Any questions about genes and evolution? All right, that's it for chapter 8. Let's just barely get started. I think it's chapter 14. Let me save that. Chapter 14. All right, chapter 14 is talking about the principles of disease and epidemiology. As always, I give you a slide giving you the major goals and a rough outline. Know the terms for this chapter. Once you know all of the terms for this chapter, and you can find all the terms in the objectives. If you know all the terms for this chapter, you will actually know 50% of the material for this chapter, meaning this chapter is very heavy on giving you a term to explain something. You should understand the relationship of the normal microbiota and its host, understand the stages of disease development, and understand what nosocomial infections are and how to control them. We won't talk about this today, but we will in the future. And let me say that nosocomial infections might be the most important thing you can learn in microbiology if you go on and work in the healthcare industry. All right, any questions about our goals and our rough outline? Pathology, infection, and disease. Pathology is the study of disease. Etiology is the study of the cause of a disease. For example, the etiologic agent of anthrax is Bacillus anthracis. Pathogenesis is the development of a disease. So know these first three terms, and obviously we'll learn a bunch of terms in this chapter. Infection is colonization of the body by pathogens or the presence of microbes where they are not normally found. For example, Staphylococcus epidermidis is all over the surface of my skin. If I were to get a puncture wound, which were to put Staphylococcus epidermidis below my skin, it would cause an infection because Staphylococcus epidermidis is not normally found there. A disease is an abnormal state in which the body is not functioning normally. When we're talking about diseases, you should realize that infectious diseases are caused by microorganisms, but few microorganisms are pathogenic. Very few of them cause disease. Most of them do not cause disease, and some microorganisms are in it, even benefit the host. Any questions about any of that? All right. The normal microbiota and the host. The normal microbiota is a very large community of microbes found in association with all animals and plants. So when we talk about the normal microbiota, the best way to do it is to say the human normal microbiota when we're talking about the normal microbiota of humans. And then when you're talking about like the normal microbiota of cats, you talk about Felis domesticus normal microbiota or the normal microbiota of cats. Those two are separate because different organisms will be part of the normal microbiota of each species. For example, humans do not normally have Bartonella henselae, a bacteria which is a human pathogen causing cat scratch fever, but this bacteria is a normal microbiota for cats. Okay, my point is just that the normal microbiota of different animals can differ. The normal microbiota are microbes that permanently or close to permanently colonize a host. For example, most of us, when we were born, picked up Staphylococcus epidermidis, And we will continue to have Staphylococcus epidermidis until the day we die, assuming that you're not, something where your entire skin is essentially sterilized. The point is the Staphylococcus epidermidis was picked up, and generally the normal microbiota will continue with us for all of our life. The normal microbiota is found only in certain places of the body. Disruption of the normal microbiota and the host balance appears to be associated with disease and impaired immune function. One example of that is when you're giving antibiotics, it kills off the normal microbiota in your gut. Maybe not all of them, but a large normal number of them. That tends to cause either diarrhea or constipation. It doesn't always happen, but it frequently happens. I don't know if it's the majority of the cases or not, but it happens in a significant number of people. And that's just the disruption of the normal microbiota by the antibiotics. Fortunately, the host normal microbiota reestablishes a balance, and then usually the diarrhea or the constipation go away in a day or two. Any questions about any of this? If not, I think I'll stop here, and I'll see you in the lab at 6:30. Is that right? Nobody's confirming it. Yeah, I'll see you in the lab at 6:30.